Hi, I'm Jordan Grant, and my lab partner is Julia Harrison behind the camera. This is the Osmosis and Diffusion Lab. We started by filling 2 beakers with distilled water and adding iodine solution to the beaker beaker A with iodine, and made sure that it was a distinct yellow color. We got 2 dialysis tubing, which are made of cellulose, which is a very thick substance that we cannot digest. And we filled one with a glucose solution and the other with a starch solution, which in the beginning was a chalky white color. We masked them before we put them in the solution, and this one was 11, the starch was 11.93, and the glucose was 13.52. We added the glucose to the distilled water, and we added the starch to the iodine distilled water. We left them for 20 minutes, and when we came back and looked at it, the starch was a dark black or blue color. The reason is because the iodine in the solution went through the membrane or the tubing cellulose, because it was small and the starch couldn't exit, so the only way to get them to diffuse was for the iodine to go through the membrane. We took the reading of the distilled water before, and it was negative, as you can see on the readings of the urine test strips. And then 20 minutes later, we took a reading, and it was pretty much the same reading, only a little darker, which means that only a little of the glucose had diffused out of the bag and into the solution. We left them overnight, and when we came back, we masked them, and the mass of the starch was the same. And we forgot, unfortunately, to mask the glucose before taking the readings, so that would be a source of error. And we took tests of the glucose outside and inside the bag, and they are very similar, which means that the diffusion was equally distributed, the glucose between the bag and the solution. When we did this lab, we should have been sure to mask before and after the experiment, and probably should have masked after 20 minutes. But we did not. And at the end of the experiment, we found that we learned that diffusion, starch is too big to diffuse through the membrane, so that is why the iodine went through, and that glucose is small enough to diffuse through, and it made a sort of equilibrium between the outside and the inside of the membrane, which means that the glucose can move freely through the membrane whenever it wants. And